students, now study biology practical question number 4 spot B, that is meiosis. Weightage of this spot is 1 mark. Okay, in this meiosis any one chart will be kept in front of you. Here meiosis, you already know this meiosis consists of two phases, meiosis 1 and meiosis 2. Meiosis 1 is again divided into 4 parts: prophase 1, metaphase 1, anaphase 1 and telophase 1. Meiosis 2 is also divided into 4 parts: prophase 2, metaphase 2, anaphase 2, telophase 2. Okay, so meiosis is completed in 2 phases. First, this is single cell. When it undergoes meiosis, first one cell gives rise to 2 daughter cell. This is called as meiosis 1. When again division is taking place, this cell gives rise to 2 daughter cell and this another cell gives rise to another 2 daughter cell, and this division is called as meiosis 2. So this whole is called as meiosis. Meiosis is completed in 2 phases, meiosis 1 and meiosis 2. Now meiosis 1, we are first proceeding with meiosis 1. Meiosis 1 is divided into 4 phases: prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 1 is quite lengthy phase. It is again divided into liptotine, zygotine, pachytine, diplotine, diakinesis. Okay, prophase is the lengthy phase. It is again subdivided into liptotine, zygotine, pachytine, diplotine, diakinesis. So at the time of exam, from this prophase, liptotine, zygotine, metaphase, prophase 2, from this everything, any one chart will be kept in front of you for your identification and for writing the comment. Okay, so let's start with meiosis 1 first. In meiosis 1, the first phase is prophase 1. If this type of chart is kept in front of you, your first point will be to identify this chart. This chart, suppose this chart is of liptotine, then your first point will be the given spotting is of liptotine of meiosis 1. Okay, if this type of chart is kept, you have to write the given spotting is of liptotine of meiosis 1. Now see what changes are taking place here. Here inside this chromatin network get condensed. Okay, chromatin network means the proteins and the DNA, everything, they get condensed. And the shape of the chromosome, how the chromosome appears, the chromosome appears to be long and thin. Whatever is visible in this diagram, those many points you are supposed to write. First of all, the chromatin network get condensed. And what is the shape of chromosome over here? Chromosome appears to be long and thin. What about the centrosome? Centrosome, which is already divided in S phase of interphase, it is duplicated already in S phase. It starts migrating towards the opposite pole. It starts migrating towards the opposite pole. So this is all about liptotine. Now we'll proceed towards another step, that is zygote of prophase 1. This is the second phase. If this type of diagram is there in front of you, if you can see the pairing of homologous chromosome here, it means this stage is zygote of meiosis 1. Okay, so your first point will be of identification. What you will write here, the given spotting is of zygote of meiosis 1. Okay, what is happening over here? What is visible from this chart? The homologous chromosome, one from paternal side and another from maternal side, these two chromosomes, homologous chromosomes, they form pairing. Okay, this pairing is called as synapsis, and these two chromosomes, this one chromosome and this another chromosome, is called as bivalent. Now these two chromosomes, homologous chromosomes, are joined together, or they are in close contact with each other with the help of one complex. That complex is called as synaptonymal complex. With the help of this only synaptonymal complex, both the homologous chromosome are in contact with each other, and this pairing is called as synapsis. And the paired chromosome, this chromosome and this chromosome, they are called as bivalent. Now what about centrosome? Centrosome, which had already started migrating, here it continues to migrate. It continues to migrate or move towards the opposite pole. So here we have completed about zygote. Now we'll proceed towards next step, that is pagete. Now see how you will identify this step. When this type of chart is kept in front of you, pagete means you have to pay attention towards this. When the overlapping takes place, what happens in pagete? Overlapping takes place. If the overlapping is visible, it means this spot is of pagete. Okay, so when you are writing this, first point is of identification. How you will identify? What you will write? The given spotting is of pagete of Meiosis One. This will be your first point, identification. Here only you will score half mark. Then what comment you will write? Here the homologous chromosome. This chromosome, this is one chromosome, this is another chromosome. Homologous chromosome, actually the non-sister chromatid. Non-sister chromatid means this chromatid and this chromatid. These are called as non-sister chromatids. These non-sister chromatids undergo overlapping or crossing over. Okay, they undergo coiling and they exchange the genetic material. Can you see in this diagram? In this diagram, the yellow part of this chromosome is coming here and blue part of this chromosome is coming here. Means in pagete, these non-sister chromatids undergo overlapping or they undergo crossing over and they exchange their genetic material. This is the most important point. Then what about centrosome? Centrosome, again their migration is continuous. Centrosome continues to migrate towards the opposite pole. So here we have completed pagete. Now coming to diplotin. Again, how you will identify diplotin? Diplotin means here now see genetic material is exchanged. When the chromosome try to separate or get repel from each other, only at certain points it is attached. Okay. Otherwise, genetic material had exchanged. Now the chromosome, both the homologous chromosomes start separating from each other, and still at certain points when they are in contact with each other, okay, then it is called as diplotin. Okay, now see what happens. Your first point will be the given spotting is of diplotin of Meiosis One. How you will identify? When both the chromosomes repel from each other, but at certain point they are coiled, then that stage is called as diplotin. Now see what happens here. Homologous chromosome, after exchange of genetic material, they start repelling from each other. Okay, repelling is started. The point where they overlap each other, certain points are there. Suppose this point is there where they still overlap each other. That point is called as chiasma, or it is also called as chiasmata. What about centrosome? Centrosome again continues to migrate. They will come just near to the opposite pole. Their migration is continued. Okay, so here we have completed about diplotin. Then last is diakinesis. Now diakinesis, how you will identify that this chart is of diakinesis? See, everything is getting separated over here. Both the homologous chromosome are fully separated. Only at the end, only at the termination point they are in contact with each other. Means this diagram is of, or this chart is of diakinesis. So when you are writing, your first point will be the given spotting is of diakinesis of prophase one. Diakinesis means both the homologous chromosomes are totally separated, only they are attached at the termination point. Chromosome gets separated from each other. Maximum part of the chromosome get separated. Only at the terminal end they are in contact with each other. Here at the terminal end only they are at contact with each other. Hence that is called as terminalization. Okay. Then what about centrosome? Centrosome reaches to the opposite pole and it start giving rise to spindle fibers. Centrosome reaches to the opposite pole and it gives rise to the spindle fibers. And in this stage, the nuclear membrane and nucleolus starts disappearing, and they disappear completely at the end. Okay, why they disappear? Because now they should give the way to the spindle fibers to come. So in order to do not create any type of disturbance in the formation of spindle fibers, the nuclear membrane and nucleolus completely disappear. So here we have completed prophase 1 up to diakinesis. Now coming to metaphase 1. Metaphase 1, how you will identify this? When you see all the chromosomes arrange themselves on the middle or at the center or at the equator, then it is called as metaphase. What will be your points? First point will be naturally of identification. The given spotting is of metaphase, where chromosomes arrange themselves on the middle or on the equator of the cell. Okay, now see what is happening here. Here chromosomes arrange themselves on the middle or on the equator of the cell. What about nuclear membrane and nucleolus? Naturally spindle formation is taking place. So nuclear membrane and nucleolus completely disappear. Now centrosome. Centrosome gives rise to two types of fibers. The fiber which is arising from one pole and coming up to the another pole, that is called as interpolar fiber. The fiber which is arising from one pole and it is coming up to the another pole, that is called as interpolar fiber. And the fiber which arises from one pole and coming up to the chromosome only. The fiber which is arising from one pole and coming up to the chromosome only, that is called as chromosomal fiber. So here we have completed about metaphase 1. Then anaphase. Now see what happens in anaphase, how you will identify. Anaphase 1 means the half number of chromosome get pulled towards one pole, half number of chromosome get pulled towards another pole. If in a cell 46 chromosomes are there, 23 are pulled towards one pole, 23 are pulled towards another pole. Okay, so let's start with anaphase 1. First point will be your identification. The given spotting is of anaphase 1. Okay, how you will identify? When you see half of numbers get pulled towards the opposite pole. Now see what happens. Why they get pulled? Due to continuous contraction of chromosomal fiber. Chromosomal fiber means the fiber which arises from the centrosome and coming up to the chromosome, that is called as chromosomal fiber. Due to continuous contraction of this chromosomal fiber, the half number of chromosome get pulled towards one pole, half number of chromosome get pulled towards another pole. Okay, this is all about anaphase. Now coming to last phase, that is telophase. How you will identify? This is the diagram for telophase. When there is formation of two nuclei. From one nuclei, when two nuclei are formed, that is nothing but telophase. Okay, this is your identification. What you will write? What is your first point? The given spotting is of telophase 1. When you see two nuclei present in a cell, then it is called as telophase 1. So what you will be your first point? The given spotting is of telophase 1. What happens here? Half number of chromosomes are gathered at one pole. Suppose here 46, out of 46, 23, half number of chromosome gathered towards one pole and remaining half number of chromosome get gathered towards opposite pole. Okay, then what happens here? Nuclear membrane get formed around the group of chromosome at opposite poles. When the these chromosomes get gathered, nuclear membrane formation takes place around the group of chromosome. Nuclear membrane arises. Then as soon as there is a formation of nuclear membrane, inside each nuclei, nucleolus reappear. Inside each nuclei, nucleolus reappear. And now there is no use of spindle fibers. Okay, so spindle fibers completely disappears. They get degenerated because the work of spindle fiber is over. So here we have completed telophase. Now coming to cytokinesis. Cyto means cytoplasm and kinesis means division. Now see what happens if this type of diagram is there in front of you. Okay, and separation, if you see that the two cells are getting separated from each other, it means it is cytokinesis. So your first point will be the given spotting is of cytokinesis 1. If you are identifying this, you will get a half mark there. Okay, now you should write what is cytokinesis. You can write cyto means cytoplasm and kinesis means division. So cytokinesis is nothing but division of cytoplasm. Now see what happens. In animal cell it is different and in plant cell it is different. In animal cells, the furrowing or the notch development takes place in the cell wall. Notch development or the furrow development takes place in the cell wall, and this furrow exceeds from periphery towards the center, like this, and finally it separates two cells, or both the cells get separated from each other. In this way, because of cell wall notch development in the cell wall, the cytoplasm divides and results into the formation of two daughter cells. Now in case of plant cell, no furrowing takes place here, okay, but actually what happens, in between two nuclei hard cell plate formation takes place at the center, which proceeds from center towards the periphery, okay, and finally it results into the separation of two daughter cells. So in this way, from one cell, two daughter cells are formed in animal cell by furrowing, and in plant cell by the formation of cell plate. So here we have completed cytokinesis. Now see, here at a glance you can see what type of structure will be there. Okay, meiosis is completed in two phases. First one is called as prophase one, metaphase one, prophase one, metaphase one, anaphase one, telophase one. Here it is about meiosis 2. When two cells are there on the chart, then you have to identify that this will be of prophase 2, metaphase 2, anaphase 2 and telophase 2. In prophase 1, what is happening? The chromatin network is getting condensed, nuclear membrane, nucleolar starts disappearing. In metaphase 1, the chromosomes arrange themselves on the equator. What happens in anaphase 1? Half number of chromosome pull towards one pole, half number of chromosome pulls towards another pole. What happens in last phase, that is telophase? Two nuclei formation takes place. Okay, so this is all about meiosis 1. Now what happens in meiosis 2? How you will identify that this is of meiosis 2? At a time in one chart paper, two diagrams will be there, two cells will be there. Okay, so in first cell prophase 2, chromatin network get condensed, chromosome will appear long and thin, okay, and nuclear membrane and nucleus, you will see that they are disappearing. So if this type of chart is there, it means it is prophase 2. Metaphase 2, you will see two types of cells are there, two cells are there on the chart, okay, and if you see the chromosome arrange themselves on the equator, means this is of metaphase 2. In anaphase 2, you will again see on a chart, you will see two cells. On these two cells, you will see daughter cell formation. Here daughter cells are not forming, but here in anaphase 2, daughter cells are formed and they are deposited or get gathered towards the opposite pole. While in telophase 2, you will get, there is formation of two nuclei. In each and every cell there is presence of two cells, and in each cell there is the formation of two nuclei. If this type of diagram is there, then you can easily identify which stage is provided to you, and simultaneously you can write your points. But one thing I want to tell you, meiosis 2 is exactly similar to mitosis. Okay, so here I have completed this part of meiosis. Thank you.